Well, I'd like to start by saying thanks to Joe for organizing this. It's not often you get the organizer for a huge conference, also is your IT guy, AV guy, and everything else.
Jack of all trades.
Jack of all trades, and especially the amazing Aven Quest for Amoeba Awareness Foundation for their support of our work and also this conference. So today I'm going to try to blend a couple of stories, but the main thing I'll talk about is discovery of genetic host factors that may be associated with susceptibility to infections with Naegleria. And you already got introduced to the problems of diagnosis of primary amoebic meningoencephalitis from Ashley and others this morning. We know that the early stages of the disease can be easily confused for other infectious agents like bacterial or viral meningitis, and that typically you have to get cerebral spinal fluid from patients and then do a diagnostic test with PCR, or you might be able to find the amoeba in the cerebral spinal fluid, like in this case here. But we know that not all cases will you actually find whole amoeba in the CSF, even up until they get to the very, very severe part of the disease. So earlier diagnosis, as Ashley mentioned, is really important. And basically, we started a project with a former PhD student in my lab who's now in biopharma, and she was looking at the biology of these organisms. She got very interested in how the organisms may interact with the host. And so she went down the path of looking at these things called extracellular vesicles, which can be excreted from the host, either membrane-bound or excreted here. You can see that basically what you end up with is these small vesicles that contain a series of things like proteins and lipids, but also nucleic acids like DNA and RNA. And we know from other fields like cancer that detecting contents of EVs can actually serve as potential biomarkers for infection. Even though that's what she started trying to do, her committee said, no, just look at the basic biology of that. But we quickly went back to the idea that we really need to find something for diagnosis. And so really where we focused was on RNA. And she did very deep sequencing of RNA from the basically conditioned media. So we're looking at only RNA that's outside the parasite and what's in the media. And all the different kinds of RNA that you would expect were there. But what we really honed in on were these prevalent small RNAs. This is over 800-something thousand reads, which means these amoeba were making quite a lot of this particular small RNA. And I don't have time to go through the whole story. You can find this on eLife now. It's a reviewed preprint. It should be a final version in the next month. But what we were able to detect is by using a quantitative RT-PCR method. You can see here, if you actually look at the copy number in the media, one amoeba grown for 24 hours we couldn't detect, but only 10 amoeba grown for 24 hours we could detect that down to that level. But if we actually looked at the whole amoeba, we could easily detect reliably even one amoeba. This is not grown for 24 hours, this is just diluted down into one amoeba. And then secondarily, if we feed these over mammalian cells and they're actively eating, they actually make more of these of this small RNA than if they're just growing without host cells around them. So we got excited and we were hoping that we'd be able to detect this as a biomarker in infected animals. And the advantage of these extracellular vesicles is that they're membrane bound, so the nucleic acids are not going to be degraded very quickly like they would if they were not within the membranes. And so we took mice that had been infected and we waited till the end stage of infection, and you can see here with all these different strains, we were able to detect this small RNA1, very easily detected versus the ones with no amoeba or other controls, and small RNA2 was also prevalent, but this is the one we focused on most, the small RNA1. And really remarkably, what we were able to find is that as early as 24 hours, but reliably by 2 days, which is similar to what Ashley said when he was finding amoeba in the brain. We detect the amoeba burden, and not just in plasma, but also in urine. So we get the mice, just rub their belly a little bit, put them over a piece of parafilm, and they'll give you some pee. Not always reliably, or they may miss the parafilm, but we get that, and we can detect this in urine or plasma. So these are fluids which are much easier to grab a hold of than cerebral spinal fluid. I mean, the case in Georgia 2 years ago, this poor girl went through 3 times of they tried to get CSF, and the pressure was so high they could not get any. And so they couldn't actually do the final diagnostic till later. But notice how over time, the amount of this copy number of this small RNA goes up, and then the final we call sort of end stage is when the animals get to the point where we euthanize them because they're going to die. So we then took this, and as Ashley said, getting a hold of human cerebral spinal fluid is very difficult. We finally got Ibn Ali at the CDC to give us 15 microliters from 5 or 6 different patients, and you can see here in cerebral spinal fluid from all of the patients that he gave us, we reliably detected this small RNA1, whereas if they were infected with Balamuthia, Acanthamoeba, or uninfected, which we had lots of different controls for that, we saw no copies. So we have a sensitivity of only 35 copies of this per microliter. You can see here they're getting 5 logs difference in there. So we're very hopeful that we can turn this into an early diagnostic, but we're also using it for another reason, which I'll show you now. And invariably, when you start talking about Naegleria fowleri and the disease it causes, it always comes down to one question. Why is it that so many people are presumably exposed? Just like Ashley said, I grew up in Southeast Tennessee, snorting water all the time, falling off of skis and whatever else I was doing in the water, and didn't get infected. Maybe there's no food for the brain, eating meat, but I don't know. But basically, why is it that some people get infected and others don't, with presumably the same amount of exposure? And is it host or parasite factors that contribute? I don't have time to go into the full story, but we have a preprint out that Chris is an author on, where we first looked at the parasite side, and we tested a bunch of different strains. And what we found was that there is a big difference in virulence between different Naegleria fowleri strains. For example, a typical one that we used in the lab, NF69 and others, you can give 5,000, 10,000, or 20,000 amoeba, and the mice usually get very ill around day 10 or 12, depending on conditions. We found one, V596, that only 10 microliters containing 10 amoeba killed mice within 5 days. So it's several orders of magnitude different. So there's very different virulence, but all of these are variant. All of these are variant because they're all came from cases. So there is differences. Is it just that people that get infected with more virulent ones, or the numbers that they get infected with? We don't know that yet. But what we really wanted to get at was the host side. And we finally came up with a hypothesis that maybe there's differences in host genetics that correlate with either increased or reduced susceptibility to Naegleria fowleri infections. And the work I'm going to talk about is primarily done by Joe Danis, a PhD student in the lab. Perla was a post-bac student who's now just started her PhD program at UC Davis, and Ann, who's a parapro in our lab. So we went to something called the collaborative cross. And basically what this is, and it's actually based here at UNC, for those who are interested, they took 8 different lines of inbred mice and then crossed those back and forth over and over again so that you get complete genetic diversity between these lines. And then they took the F3 progeny from these that were all very diverse, and then started going 15 generations of inbreeding. So you develop individual CC lines, collaborative cross lines, that are genetically distinct, but all the ones in that line have almost completely identical genetics. And so then you have all these that you can then test. So our hypothesis was that if genetic diversity is involved, we could use these mice and try to identify factors that would either give increased susceptibility or refractory infections. And for this study, we had 3 quantifiable traits that we decided to go for. One are the survival curve analyses that you've heard about a little bit already today, the amoeba burden, which is our qPCR method I just mentioned, and a little bit about immune pathology. So If we look at just the founder lines of those 8, we can see right up front that there's actually differences. The AJ is much more susceptible than some of the others. And you can see here that these are more refractory. So some of these lines out to 30 days are complete survivors with exactly the same inoculum as the ones that got here. We have a lot of strains that sort of fall in the middle around what we typically found. So the next graph I'm going to show you is quite complex, but just bear with me. If you look on the left, this is the day of survival, and this is organized by the most susceptible strain to the most resistant strain of mouse. And you can see here, several of the lines are similar, and the color-coded are the founder lines, so that's the AJ, and you can see here, some lines are really hyper-susceptible. They become infected very easily and quickly, whereas others actually progress to day 30 and actually survive completely. In fact, we don't know how long they will survive because our endpoint was originally 30 days because we thought that was long enough. It looks like we could go much longer than that, and these are actual real survivors. The other thing we did was we looked at both male and female mice because there's always this idea that you see more males that get infected than females. Is that really a difference, or is it just difference in activities or exposure? And most of the data we found, it doesn't seem to be associated, but we did find one really interesting link that I didn't expect. For example, we're using a statistical method known as quantitative trait loci mapping, QTL. Basically, this is a fancy way of saying, if you look across all the genes in the genome, which ones are associated with increased survival or increased susceptibility. And so you see all these squiggly lines, which don't mean a whole lot, but then you're looking for something that crosses the barrier up here that's statistically significant. And basically, this is based on the survival data only with the first 30 lines. We're seeing a nice QTL, which is there on chromosome 8. And when you look at the genome sequence across this region where this marker is, it's right here. And you can see here, for example, AJ, which is the most susceptible, is down here, and other ones are susceptible. The most resistant ones are typically up here. So there's something in this region right here where we're finding the QTL that is associated with either resistance or refractoriness. And this also holds for, if you look at the 8 founder lines as well as the CC lines. So pretty exciting. If we look at that QTL marker, basically that means we have a number of genes in that QTL. And in this case, we found 17 protein encoding genes that are in that QTL. And of those, we don't know which ones are the most important or have a role or associated yet, but 2 popped out from just the interest level, neuropilin 1, which is regulates and interacts with regulatory T cells, and interferon regulatory factor 2, which is also in, both of these are involved in inflammation and involved in immune responses to different pathogens. So these are some promising results that we can now go back and test these and other candidates from our analyses. But I would say that basically at this point, we've only got the data for the first 30 lines, and we're fortunate to be funded for an additional 20 lines or so from the Aven Foundation so that we can have a really nice, complete story and associate those with survival curves. But we could also look at a different phenotype, and one of those is amoeba burden. As I introduced to you the idea that we could use this for diagnosis, we can also use it as how many amoeba are there in the brain as a correlate of that. And you can see my student has a nice sense of humor. He calls it qPCR since we're using urine. But it's non-invasive. So, typically before we would infect a mouse, and as Ashley said, you don't know what's happening between the time you infect and when they start showing symptoms, but we can actually monitor that. We don't do this in real time because we get lots of P samples from lots of mice at the same time, so we go back and retroactively do this. But we can now look at amoeba burden to see how that correlates. And this is still new data, so bear with me. Basically, you can see these are, sorry, make sure I say the right thing. Yeah, this is the copy number over here of that small RNA1. This is the day that they survived to. And you can see the ones that die early have high burdens of the small RNA, and then the more refractory mice have very, very low burdens, and in fact, sometimes it's zero. So here's a typical sort of survival curve in the lines of male and female mice that are infected, and then the circles are the qPCR data in copy number, and you can see that as the copy number goes up, the animals start to die. So we see the same thing for other susceptible lines in the CC. I'm going to just skip that and go to the one I think is the most interesting right now is what's going on with these mice that seem to be refractory and don't get infected or do not die of the infection. And first of all, the y-axis for number of the burden of amoeba is much lower here, but you can see here that we have evidence that these amoeba are actually crossing the blood from the nasal passages probably into the brain, and we're seeing copies, but then they're resolving over time. So it seems to be that some of the amoeba get across and then the immune system controls them or somehow. And so these are really valuable mice because we can start to understand why some amoeba are not causing disease and why some mice are actually being able to survive the disease. Of course, on the other end, it would really be helpful to understand why are some mice more susceptible. Maybe this replicates what we see in the situation I mentioned where you have 2,000 people, one person is much more susceptible for reasons we don't know yet, and others are, this may be the actual typical person is more refractory and some people are just more susceptible. So the last thing which is really interesting for us is something we observed in mice, and I apologize, this is Halloween, but if you don't want to see a brain, you can close your eyes for a minute. When you look at the mouse brain, this is the posterior brain, the frontal part of the brain, olfactory bulbs. This is where the amoeba first start to come in and do their damage, and that's indicated by the arrow here. They typically, when we only give it in one side of the nose, they typically go to the one side and cause severe hemolysis and the brain-eating aspect that we know about. But what we found is in mice that survived the infection, especially a couple of lines, we saw this really curious phenomenon. These are mice that survived to 30 days of the infection, and notice the olfactory bulbs have these dark pigmented structures here in the olfactory bulbs. And what we found was that we only see this pigmentation in mice that survived the infection compared to what we see here. And so when you look at this, histologically, you can see the melanization. These are basically melanocytes are depositing melanin mostly along the blood vessels in this part of the olfactory bulbs of the brain. Now, we do not know if the melanocytes are actually involved in the immune response that's helping protect, or maybe this is just sort of a sign of a protective immune response and then the melanocytes deposit. So trying to untangle that will be pretty exciting. But if you look at it on a statistical basis, we have seen this melanization occur in 5 different CC lines and 1 founder line. And you can see the difference between mice that have a typical susceptible response versus those that are resistant. The presence of the melanization occurs in the ones. In fact, in this CC line, 7 mice survived out of 8. All 7 survivors had this melanization. The 1 that died did not have it. So what is the role of this is really curious to us and something we're going to follow up on. So I've tried to give you a quick overview of some of the things going on in the lab. We're still involved in drug discovery and a few other things, but right now we're really trying to hone in on what are these genetic variations. And so we want to get to at least 55 different CC lines, so we have a really robust QTL analysis to identify other QTLs. We already have some indication that with the PCR data, we're finding some other QTLs that are not the same one as we found with the survival curve. Secondly, we want to map these for susceptibility and refractory responses. We want to examine the immune response, especially the melanocytes and how they're activated in this model. And then we're actually already doing this work. We've applied spatial transcriptomics, where you actually look across the brain of a mouse that's either infected or not infected and look at the host responses to the amoeba infection. And thanks to Ashley for the antibody, the green here are the amoeba. This is the frontal part of the brain going to the back. These are small areas where we can actually do the spatial transcriptomics, and we've already been able to detect differences in the expression of cells, the host cells, right in the amoeba infection versus just before it and further back. So this sort of work with the different virulent strains, as well as some of the CC mice, may also give us more information about the expression profiles of during a resistant response. And then this can't be done without a team. Joe Danis, as I said, is doing the CC project, Perla, and Mac is the QTL expert for doing the analysis. Anissa is working on drug discovery, combinations. Cassie did all the EV work. Anthony Roberto is a wizard with single-cell RNA-seq, which I didn't have time to do. And a lot of other people, including Chris and Ashley and others in the room, Ibni, and the UNC Chapel Hill group that is providing all these mice to us. They've been very, very helpful. And then all of our early work was done with money from the NIH, or the Georgia Research Alliance, but we're very happy and glad to be associated with the Aven Foundation for what we're doing now. Thank you, and I'll be glad to answer any questions. Yes. We don't see that. If you look at our previous work that Chris alluded to, you can give like 5,000 amoeba and you have some that survive and some that don't. So there's a certain number that is needed to get virulence, but then between strains, there's different virulence. So the V596, reliably with 100 amoeba or less, we get death within 5 to 6 days. Others, it takes about 2 weeks. So there's a difference there. And to answer the other part of your question, I think, is that we give the same inoculum to every mouse. So what we're doing, we do these in batches for the CC. So we always have one control line, which is our black 6 mice, so that if we see a shift one way or another because of the inoculum, we can compare between every experiment. Because each one of these experiments is a lot of work that Joe's doing just to monitor them and collect the analysis and do all the PCR data. Yeah. Okay. Well, thank you.
Which one is yours?
It should be, left it open. Oh. Here
You have to
it is, here
restart
is.
it for it to be able to work properly.
That makes sense.
All right, cool.
There you go. Hello, everyone. My name is Naegleria de Aguilar, and although I'm not doing a PhD in Naegleria right now, I used to work for the Costa Rican Water and Sewage Authority, where I actually met Joe several years ago. And there we used to be in charge of everything water related in the country. So what I'm going to present right now is how, from the perspective of a national water authority, how we approach a series of cases of Naegleria that were tied to hot springs in Costa Rica in 2020, and how the healthcare system also managed to keep on track of cases from 2020 on. Just to give a brief background, the data related to Naegleria in Latin America are scarce. There are just a few studies from Mexico and 3 or 4 studies from Costa Rica, and that's it. And at the level of water and hot springs, we do not have any quantitative data. We just have absence or presence, which is really interesting based on the fact that now molecular tools are widely available. Microbiological tools have been around for decades, but for Naegleria, this is not the case. And also, talking with colleagues from Costa Rica, they think that most of the cases that are detected are the ones that have an hospital nearby, and that we might be overlooking some cases that can be misidentified, as Dr. Gerba said, as viral meningitis or even bacterial meningitis. And why Costa Rica? Well, we in Central America emerged from a volcanic origin, so we have a bunch of hot springs from Mexico to Colombia, and specifically in Costa Rica, these 2 areas further north here, kind of like cluster all the hot springs in the country. So we have the Arenal, Miravalles, and Rincón de la Vieja volcanoes, and this attracts geotourism. So people that want to escape the winter from the Midwest and kind of want to get warm a little bit, they go down to Costa Rica and they use these hot springs. And since 2014, we have detected at least 4 PAM cases. Anecdotally, one of them survived, which unfortunately was not reported in the literature, but I'm going to talk a little bit about it later. And how do we monitor water in Costa Rica? Well, everything is kind of centralized regarding drinking water. So there is an authority in charge of actively monitoring for indicators and pathogens. But when we talk about other types of water systems, including seawater, lakes, and even hot springs, we have no clue how to approach that. And so when I used to work for the Water Authority, we did an effort. We kind of tricked our budget and started looking into other types of water systems. So we championed the wastewater surveillance system. We started looking for pathogens in seawater, and we kind of were interested in hot springs. We never actually started working in hot springs until 2020, when there were 3 cases detected of Naegleria in the country. 2 of these were epidemiologically tied to hot springs. One of those are suspicious to be tied from a groundwater source. And this 5-year-old survived. I was talking to Dr. Gerba yesterday. This was a pediatrician intuition. The parents of this kid took her to the doctor, and that doctor immediately started antimicrobials without having any simple taking. And so it's unfortunate this is not in the literature. I think it's one of those handful cases out there that actually survived. And so we were called by the Ministry of Health, and they said, we have had 3 cases in less than 6 months. What can I do? How can we approach this? By that time, we have approaches already established for viruses and bacteria in different water systems. And so that's what we did in the field. We went to the field and deployed several systems that we have, including kind of like viral based approaches, such as the viral method using electropositive charges, charge-baked cartridges, and whole fiber ultrafiltration, and also membrane filtration. We didn't have time to have a quantitative positive control, and so we ended up doing absence and presence of Naegleria. And we ended up detecting this pathogen in 2 hot springs that were actually related to volcanic geothermal systems. So we were also wondering whether this is a factor or not for the occurrence of this pathogen geographically, at least in Central America. Fortunately, these findings were corroborated by an independent research group at the University of Costa Rica. They found the epidemiological link between the cases, the water, and they were able to even isolate that Naegleria from both cases. And just to give an example of how much we need quantitative tools, they still use regular or conventional PCR to quantify, to detect Naegleria in water. So this faint line that you can see here on line 7 is actually the Naegleria that was isolated from water. So what happened after this is that the Ministry of Health kind of was more aware of what was going on. It was only 4 cases since 2014, so they started a signaling campaign. So this warning signal regarding this disease. The problem here is that geotourism was heavily affected. And so all the geothermal results surrounding this volcano saw a reduction of around 80% of visits in comparison with the previous 2 years. And so they were calling us, kind of blaming us for what we presented in the news, which was really conflicting. We were just spreading awareness. And so, yeah, that was kind of an issue. This is the sign this is the same sign. I actually found this on Reddit. So people were, there is a Costa Rican thread on Reddit, and people were sharing this, kind of creating awareness, but also scared. Some of them had no idea that this was an issue. The same sign was shared across social media. Of course, all Facebook was flooded with the same sign, and of course the tourist people were heavily affected. They also enforce using chlorination in the hot springs, which can be an issue because we still don't really know how well chlorination works in temperate waters and against Naegleria, and I think that's part of the work that you're going to do at Purdue. I think that's amazing, and we're going to show some light regarding this. And the same group that I just presented did a social environmental impact work that was published a couple of months ago. They went and sampled across all the entire this volcano area that I just presented, and they tested only 22 sites. 7 out of those 22 came positive for Naegleria, but they came with this approach of doing surveys on the field. And what they observed is that around 40% of people that live on the nearby of these ponds and that use the ponds regularly for recreation had no idea what Naegleria was. And the way it works is that these ponds are usually in sort of rivers or springs, and so the private owners will take a chunk of this water, create a resort with a government kind of concession, but then the other part of the water is for public recreation. And so if you drive across this river, you will see dozens of people enjoying the water without knowing about this risk. And the signaling campaign, unfortunately, was only done in private resorts. So I just want to finish up with some thoughts about what we were talking in Costa Rica before I came to the States and started graduate school. We had no idea how to approach quantitative assessment in terms of in situ assessment, how we can get really good numbers on the field and provide even the resort owners with this data. We don't know about seasonality and geographic distribution. We have a few lakes, but a lot of hot springs, and I think most of the literature have reported numbers on lakes, and some of those reports are also in rivers. We have hot springs, and that's why we need the data right now. There is virtually no QMRA approaches for hot springs. And that's something we are missing. The controller management right now is just chlorination and signaling. That's it. We don't know what to do. Some of the water slides were prohibited, and I think that should have been done way before the cases. And we pivoted this idea of in lower middle income countries, it's really difficult to go to the field and start looking for all the pathogens at the same time. We might be lucky here that we have the resources to do multiplex or even tag arrays. We don't have that in lower middle income countries, so we relied on indicators. For fecal pathogens, we have a bunch of indicators. For viral pathogens, we have indicators. But what about for free-living amoeba? We have no indicators. And so we pivoted some ideas of how to mining these indicators, including looking for relationship between bacteria that these amoeba are feeding from in the water. Are they feeding from specific bacteria? Do they have any sort of preference in that terms? And with now the search of machine learning AI, can we come up with models that will predict how this amoeba will grow in a certain environment? And we engineers love mesocosm and microcosm. So can we come up with mesocosm experiments that then confirm what these models are showing us? And so we can come up with better data on persistence and fate and feed up QMRA approaches. And finally, and this is very ambitious, functional interactions. Can we include metabolomics here and see whether or not the nature of the water can influence the pathogenicity of this amoeba once they infect a human. That's what I have for today. I'm really happy to address any questions. I would like to especially thank Joe and the Aven's Quest for Amoeba Awareness for having all us here. And I want to thank the graduate school for the grant they gave me to fly to Chapel Hill, and to the people that I work with at the Water and Sewage Authority back home in Costa Rica. It was a wonderful pleasure to work with them and to create some new knowledge. And yeah, I really appreciate it.
So in your hot springs,
Yeah.
do you have other water quality measurements for those? So could you go back and look at the iron levels in those to see if there's a elevated in those particular hot springs
Well, we have pretty much what they have to report to the Ministry of Health:
on their iron level?
pH,
Okay.
turbidity, and fecal coliforms. That's it. So we are looking for, we're using an indicator for fecal pathogens, and fecal pathogens might not be the problem here. So, yeah.
In terms of inactivating Naegleria species in hot springs as an alternative to chlorine, have you thought about looking at copper? I mentioned because there was a student doing some preliminary work with Naegleria grouperi, and they got pretty decent inactivation with about 1 part per million copper. And so I was just curious, it's something that might, last a little longer than chlorine.
No,
Whether you
yeah.
want to consider that in like a head-to-head.
No, I think that's an amazing idea. By the time we never looked into that, and I think I'm gonna spread the word definitely and share this with them. But yeah, I appreciate that, Mike.
I mean, this was just grouperi, not pathogenic surgeon, so just
No,
something to look at.
but 100%. Yeah, thank you.
Can you comment about what the temperature variation you're seeing and where they're isolated from the hot springs? Because we can grow these out at 44, 45 degrees C, which is usually hotter than anything we actually
Mm-hmm.
uh, was that part of your measurements or the data?
Yeah, we were never able to isolate. We were not trying to isolate it, but this independent research group were able to isolate the Naegleria at 45 Celsius, and I think they reported around 43 to 47, the range of temperatures when they were actually sampling. Whether or not that changes throughout the year, I'm unaware, but I think it was an average of 45 Celsius for those hot springs.
So you mentioned 80% reduction in people going into the water. Obviously, this was 2020 when COVID-19 was spiking and there was a reduction in people traveling. Did you ever counter for that? And was it truly 80% because of some pictures of Naegleria?
That's,
or was it
I think that's a great confounding factor. Yeah, we didn't do it like a statistical study that, to properly look at that. It was more communication with the resort owners. And what we heard, or what I heard later, is that it kind of happened what happened with like mask usage. It kind of spiked in 2020, and in this case, like the reduction of visiting declined during all 2020, but then it spiked again in 2021 because people forgot about it. It causes this shock in the news, so people will avoid it, and then when kind of everything settles down, people forget about it and will start visiting again. So I think it's important to at least have some practice and some signaling in place to kind of remind people that this is a problem. But yeah, it's an important confounding factor that could have played a big role.
Because there's a couple of places here that
Yeah.
have signs, and there's still people jumping in and out of ponds and lakes, you know.
Yeah,
I wonder
100%.
how
Yeah,
efficient those signs that warn people and how trusting that
Yeah.
those people actually take those signs, you know.
thank you for that, yeah.
Okay, we're doing good on time. Thanks, Andre. What Andre didn't say is he's starting a postdoc here with me in January. So Andre, you're about to learn about what your project is going to be on. But green light on the indicators work. I think that's a fantastic idea, actually, as well. Let's see. Yeah, absolutely. Let's see if I can find, oh, here we are. There's so many interesting things to talk about. I'm just really grateful to everybody who's come here and for doing this. My mind is just racing with all the possibilities. So I'm really interested in advancing environmental monitoring because, as I think we've seen throughout all the presentations, that we're really in a data scarce space here, right? We don't know that much about Naegleria ecology. We know these factors may be copper, maybe iron, nutrients, specific bacteria that may be driving Naegleria, when and where Naegleria exists. And my whole career has really been built on developing new methods for generating additional data so we can use data in better decision making. So I just want to start out with some thank yous rather than leaving them to the end. So thank you to the amazing Aven's Quest. And thank you to some of the people in the room. I'll just have you raise your hand. Frederick's there. Frederick's done a lot of the work that you'll see. Gunho Lee, Kevin Zhu, who was with us until recently, Megan Lott in the back, Timothy, and Mike Fisher back there is also part of this project. So I want to start out with just a few observations. I mean, one is, as we've seen, Naegleria is relatively common in warm recreational waters. Like if you go out and take samples in hot springs and lakes and ponds, you'll find it. It's there. It's around. But cases are extremely rare, black swan events. Having said that, there are some places that have experienced multiple cases, right? And that suggests to me that there are specific times and places when you might see elevated Naegleria where the risks may be greater. So it's more than just individual susceptibility. I believe it's also environmental specificity in terms of the distribution of Naegleria. But we get this question a lot: is routine testing appropriate? Are we going to scare all the tourists away, for something that's very rare, or should we, get out in front of concentrations of Naegleria when we can? My hypothesis is that more data on when and where Naegleria exists and where it's elevated, especially, can inform risk mitigation. And so our goal is really a rapid, scalable method for environmental surveillance of Naegleria. And that's because there is no rapid field-based method for Naegleria fowleri measurement. So my whole thing is we need more data to be more useful in any approach to prevention, which is, I think, very important. So I want to just zoom out a bit and say something about how we look for microbes in water. It's a needle in a haystack problem, and I want you to just keep that in mind. It's a very challenging issue, because the relevant levels, and I'll get into some of the monitoring targets that you would want to know about, are vanishingly small. And Naegleria exists not only in water that's complicated, there are particles, there are chemicals, but there are lots of other microbes in most waters. So you're looking for, let's just say you're looking for a coffee bean in a volume that's the equivalent of an Olympic-sized swimming pool that's also filled with other coffee beans, but that are not the ones you're looking for, right? So it's a very big challenge. So say you have something you want to look for in water. Generally speaking, one way that we do this is we filter to concentrate. So you're trying to take a big volume, filter down to a small volume that you can look at analytically. You want to be able to measure the volume that flows through your filter. Then you want to be able to take that filter, extract microbes from it, look at their DNA typically, and maybe get a positive, negative. Maybe you get quantitative data. Maybe you're able to, depending on how you've captured these things and concentrated them, maybe you're able to culture them, which provides you with a lot more and better information around potential exposure risk, since DNA doesn't tell you anything about viability, obviously. And we do this for lots of different microbes, and we do this in lots of different ways. Some of the existing methods, there are lots of methods that are out there that have been published for detecting Naegleria in water. All of them are laboratory-based, and they're generally only used to confirm the presence of Naegleria when a case has been reported. So there's very little prospective monitoring, right? Because it's very difficult to measure these things, and it's usually a reactive process, right? And I don't expect you to read all this table, but there are lots of methods, including, I'll pick out bits and pieces of this as we go along. Looking at PCR sequencing, using different PCR approaches, nested PCR, which somebody mentioned, LAMP, which we're using in one of our analytical workflows. Culture and qPCR tends to be for the places that do have monitoring regulations, like Australia. It's a culture plus PCR method. That's recommended in France as well. Different sequencing methods. All of these are expensive. They require specialized equipment, and they take multiple days typically. And including with culturing, it might be several days to culture and then PCR confirm what you've measured. So there are a lot of things on the table. I think this is a sandbox we're playing in. So we're trying to take bits and pieces of this and really look at it through the frame of what's possible to do quickly, relatively cheaply, and basically in the field. But before I get into that, I'll just ask sort of what the target is, right? So you can develop a method to measure something, but measuring something that has a very low limit of detection is going to be very difficult typically. So it's good to define a priori before you move into work like this, like what are we trying to measure, right? What's the use case? Are we trying to distinguish between any Naegleria at all versus none? Are we trying to just detect really high levels? Because we might use that in our prospective testing. So when you look at cases, and this is also, people have mentioned parts of this as well, and I want to credit Lyndon over here. Raise your hand, Lyndon. Lyndon's been helping us do some of the risk analysis and calculation behind this. But if you look back at cases, and there are not that many cases that also have relatively simultaneous measures or near-time measures of Naegleria in water, you can see that there is really no pattern, right? There are cases that have been identified, and then there's been retrospective sampling that shows very high concentrations and others that show pretty low concentrations. So there seems to me to be no real pattern, but it's very data scarce. Then you can also look at a QMRA approach. Andre mentioned QMRA, quantitative microbial risk assessment. This is when we take dose response models, and for Naegleria we have mouse dose response models that have been very well developed. And build off the quantitative relationship between dose or concentration in water and probability of infection or probability of death. So what you can do is, and I think Chuck may have mentioned something about this too, you can take a target, a risk target, and you can say 1 in 10,000 risk of infection, or typically we use something called disability adjusted life years, which is a measure of both morbidity and mortality that's used in setting health-based targets for exposure, to back calculate a risk of infection and then a dose and then set a target that way. So you can do this, you can calculate this. When we do this, however, using the mouse dose response models, which are very highly conservative, you would come up with a target of something like 3 × 10⁻⁴ Naegleria per liter, which is not a realistic target. We can't measure down to that low concentration, number one. But number two, that can't be true for humans because we know that that Naegleria exists at much higher concentrations and that not everybody is getting sick from it. By the way, there's a QR code here for Lyndon's calculations around risk if you're interested in that. I'll let you take that picture, Mike, before I continue. Now, the one sort of risk-based standard that's been developed is for France. So France does have a risk-based standard for their monitoring. And it's this paper published in Applied Environmental Micro several years ago that took some of the dose response models from mouse models, but also brought in a probabilistic assessment of how people might be exposed, including what the exposure might be through nasal exposure, to look at the risk of PAM and how that relates to potential Naegleria concentration in water. And I won't go through their entire analysis, but just to say they use this to set a threshold, a risk threshold of 100 per liter as a probably good enough estimate for setting kind of health-based monitoring going forward. So they've done that. And zooming out, I mean, there are different ways that we could look at setting targets for monitoring, right? So Australia actually has a non-detection threshold. So less than 2 viable organisms per liter is what they recommend. France has this 100 organisms per liter based on their risk calculations. There's the QMRA-based risk calculation, which is very, very conservative. There are other approaches too for recommendation of recreational waters in Naegleria, including looking at temperature proxies, because we know this is a thermophilic organism, to avoid swimming in waters that are of higher temperatures. This is really hard to do, it's impossible. Or other microbial proxies as well, and this is the indicator idea that Andre mentioned. But we've decided, based on all these different approaches, which are all flawed, that we would like to set 100 per liter as our initial target for monitoring, which is still very, very, very challenging for reasons that I'll get into. So some options here. These are the different steps in that cartoon that I showed initially. So we typically need to concentrate large volumes of water. Concentrate, we need to elute, so that's you concentrate on a filter, then elution is removal of that material from a filter. There are various methods for that. Processing, so in our case, we're looking at molecular methods, extraction of the DNA. Application of IMS is also used to get really sensitive detection of Naegleria. Usually not going to be field doable, and also requires the antibodies, which are hard to come by, and we should talk about that, Ashley. And then detection. So detecting the thing that you're looking for requires either observing it directly by a microscopy, which can't be done in the field necessarily, and can't be done on low concentration samples, culturing it, which takes a long time, sort of standard qPCR, digital PCR, which requires specialized equipment in labs, same with nested PCR, LAMP, nanopore sequencing, aptamer-based assays, or there are other things as well, and we can get into those if you'd like. We've settled on, for our rapid method, using a filtration method, which we'll talk about some of those in a second, and LAMP. So we're using this loop-mediated isothermal PCR method, which is field doable. It's pretty rapid, and I'll show you pictures of that in a minute. I think there's a lot of potential in using aptamers as well, and protein targets that might be easier to detect. So in this work, we're always looking at trade-offs in terms of complexity, time to result, cost, and lower limits of detection. And we've sort of defined for ourselves that this has got to be workable in the field, right? By the way, this presentation is more about the problems and the challenges than the solutions, because I'm not saying that we have an answer to all these things. I'm just saying I'm trying to explain kind of why our approach is the way it is. So for LAMP, this ends up being a presence-absence assay that targets this virulence protein and is a 1-hour incubation with a 2 microliter analytical volume and pre-made master mix. So we're building off a previously reported assay from 2015 that actually has a pretty low limit of detection when it's coupled with EPA method 1623, which is the method that we use for identifying crypto and giardia in water. Very laboratory intensive filtration and immunomagnetic separation, which we can't do that part. But the way it looks is you have this chromogenic assay. This is before and after. I don't know if you can see that there's a color change involved between positive and negative. So it's something that can be done in the field to detect genomic DNA, a gene target that's specific to Naegleria fowleri that changes color. And also we're doing a lot of digital PCR as confirmatory testing. So both for fowleri and grouperi. So we're taking qPCR assays that have been published and transferring them to digital PCR, which is much more sensitive, that we use in my lab. And the output looks like this. It's like you're doing many thousands of presence-absence assays. This is fluorescence intensity, where you can see positive bands versus negative bands that are thresholded. So the two options that we're currently working on, we refer to these as the jetpack and the UFO. I don't know why. Do you know why? I don't know. Anyway, we'll get into that. The jetpack is one method. This is kind of an off-the-shelf eDNA device. So eDNA measurements in the environment have been developed for things like assaying environmental media for rare species, right? And it's this thing right here. This is a filtration. There's a pump in here, a flow meter, a pressure gauge, lots of other things. And it works with this sort of filtration apparatus out here that has, it's got 3 different filters on it. And you can stick that out there, and it pumps water through a membrane, either 3 or 5 micron pore size, and then can be used as the basis for elution, extraction, and detection, right? So this thing, has some strengths. It has a, relatively low flow rate and pressure, which is good because we found that Naegleria can be sucked through these membranes and destroyed. We like to collect them on the membranes. There's built-in sensors, data tracking. The small filters are easy to work with, but it's a pretty low volume. And again, this is a needle in a haystack problem, so typically we want very large volumes. It can clog, and there's pretty low recovery we're now seeing with these. So in terms of if you spike in, and in the lab we do experiments where we spike in Naegleria grouperi and then look at recovery, it looks like these are pretty low recovery, unfortunately. It's about $25 per assay, including the lamp. So again, one of the other things we're looking at, not only like limit of detection, time to result, is also cost, because anything that's going to be scalable, we want it to be not that expensive. So the way this works is water gets sucked in here, and it comes out over here. It's retained on filters. We are sampling around here right now, so we have 6 sites that we use, and we're looking at differences in filters. University Lake down here, there's Lake Ellen, Anderson Park, and then there are 2 kind of farm ponds up here. One of them is mine on the 50 acres that I recently bought as a midlife crisis. And I'm very interested in it. The other one is my wife's uncle's pond that we go swimming in all the time. So I have a personal interest in these spaces. Here's Frederick out sampling. This is me just performatively standing there because he's doing all the work. This is my pond. Something we've done is, again, we spike in grouperi in here, and we look at the recovery on the filter on different types. We also do this with, we've done this with our positive control for fowleri as well, to look at retention. And typically we'll take the filter, divide it in half, put it in tubes, do the bead beating step, and then take that out and do dPCR and lamp on what comes out. So this is what the dPCR output looks like. We were getting like low recoveries for the jetpack, something like 6%, which is not good. So we're trying to work on methods of increasing that. And again, we're working with grouperi in the lab. These are some cysts and trophs that we that Fred has spent a lot of time trying to grow. We have actually not detected any fowleri in any of our samples yet from any of those 6 places. So we started sampling in September, October, so it's getting cold. It's been relatively cool here for a while. So I don't know that I'm super surprised based on that, but we're going to carry on field sampling. And then our other filtration setup, the one actually we started on, we call the UFO, and I'll show you why in a second. It's a bigger filter, and we're putting a lot more water through it. So we can filter up to 100 liters within about an hour. It's simple, it's easy. It's pretty high pressure, which may also damage the trophs and suck them through the filter. And it results in a large membrane that has to be cut up and eluted from, and there's some challenges there for processing. It's about $18 per sample, including the lamp. This is what it looks like. So it's very simple. I love simplicity as an engineer. So this is a pump over here. It's a half horsepower field-ready pump. There's a pre-filter on the end of this thing. There's a filtration stage here with a pressure gauge on it and a flow meter on this side right here. And That's what it looks like in operation. That's why we call it the UFO, because it's got, it has that UFO shape to it over there. See what it looks like? And same thing, it goes through this filter and out this way. We're hopefully retaining Naegleria on that filter. We do turbidity testing, and you can see, you can get a cake that's on here. This material is what we're going to be interrogating for the presence of Naegleria, obviously. That's us measuring Gruberi in the lab. So some next steps, we're doing further recovering and field testing of these things, including UFO recovery testing. We don't have good recovery data for the UFO yet. We're developing a pan-Naegleria lamp assay as well, so that we can use that on our Gruberi lab strains that we're using for the lab testing and recovery in the lab. And we're looking for alternatives to the current elution and extraction methods that are quicker, cheaper, higher recovery, more field appropriate. And my greatest interest is determining our lower limits of detection for these assays and workflows, and really working on driving that down so that we can get closer to the 100 target. We're much higher than that right now, and we need to account for a lot of variability in physical and chemical parameters in waters, because any of the waters that we might be interested in measuring as part of surveillance for this are going to have a lot of variability around the parameters. Some of that can influence the lamp assay, we know, and it can influence a lot of other things, including how much water we can get through filters. The other thing we want to do is evaluate some alternative detection methods. So we think LAMP is pretty good. It's basically the only quick field-ready PCR-based assay that we can use, but very interested in lateral flow tests, interested in these aptamers, and the potential to develop a method that can give us a better and quicker result based on the samples that we concentrate. So that's it. Thank you. Happy to take questions.
I just wondered why I never thought of using Naegleria. We always use fowleri.
Okay.
Which we had to put up signs all over about the hazard of doing that, but I never thought of that together.
Yeah, yeah.
Joe, I was curious about the actual water that you test. So I always sort of foolishly assume that if I were to go out in the middle of a large lake, there's not Naegleria swimming around in that area. So I always imagine the risk really lies in when you're stirring up
The sediment.
at the edge of the lake and then playing around there. So when you guys are testing, you reach out how far? I mean, I don't know how water sampling works, but you reach out so far, or you play around in the water a little bit and then do that? Because I'm sure you're gonna get a ton of sediment, but maybe how do you do this?
Yeah, so we're sampling right on the edge of our ponds that we're sampling. But that's a great question. I mean, I think that's one of the things that once we have a method where we can get really rapid results, then we can start to ask those questions. Is it swimming out in the middle of the lake where it's away? We can do experiments where we stir up the sediment. I mean, we know that it's to be found to a great extent in sediment. So I would assume that would have a big influence. Yeah.
Because if you say you can't find, you haven't found it yet.
Yeah.
And I guess if it's all just sort of stationary, completely settled water, I guess that's not that surprising. So I wonder, I don't know, you guys just take your first sample, stir it up a little bit, take it again, because maybe it's just there. It's
Yep.
just swimming around when you're sucking.
Yeah, especially if there's evidence that it's especially when it's cold, right? That's the environmental reservoir for it. So that's a good idea. We should try that on our next sampling run. Hmm?
Many decades ago, for my PhD, we did that, and in the middle of the lake, basically, we still found just as many amoeba on the thermocline where the temperature difference per particles actually sort of slow down, and that's where the bacteria we try this are as well. So the sampling question is what I was going to ask as well, but I think you're hitting on the same idea. Most of the amoeba are actually in the soil or the sediment. So you can actually use methods, and the French do this very well, better than I did in the old days, is actually washing that and then filtering it and looking at, you might actually increase your sensitivity by looking at sediment, even in the very shallow parts, compared
Yep.
to what you're doing in just the water.
Yeah, great. We'll do that. Yes.
Really like the idea of looking at LAMP together with LFIA, just because it seems like even if the LFIA performance is optimal, it's not going to give you the same quantitative working
Yeah, I mean, that's a great suggestion. I mean, I think we haven't really played around with anything beyond LAMP, but I think my interest is really in creating the most efficient workflow as possible, and that may include that as an option for sure. Yep.
More of a comment rather than a question. My lab's been developing a LAMP assay for ballonophilia and also a digital PCR method for ballonophilia for diagnostics.
Mhm.
So we should probably talk about trying to get it introduced so that you can also monitor for other free-living amoeba as well.
That's why we're having this meeting. Yeah. That'd be great. That would be great. Yeah, that would be great. Yeah, for sure. Yes.
Forward thinking, when you're successful with your approach, how are we going to actually increase awareness without increasing the fear of the tourism issues around different areas? Because I was in Florida many years ago and the Department of Health, and they were debating, do we put up signs or we don't put up signs?
Yeah.
They were worried if they put up a sign here, 500 yards down around the edge of the same lake, somebody goes in and gets infected and they say, you didn't tell us.
Yeah.
I mean, how are you planning to use this information in this sort of social way?
Yeah, I mean, in some ways, like, as big of a challenge as this is, really getting quick, good data on Naegleria in the environment. The harder questions are, what do you do with the data and how do you communicate it? And I guess what I was thinking about that is, once we have a way to get data and we're starting to collect data, we start the process then of talking to the public health authorities, talking to others around how you would use this in a responsible way, how you would communicate it without creating unnecessary fears, but by also describing what the risk could be. I mean, I think it's a very interesting and difficult problem. And it's not really one that I know that much about.
You know, one thing might be what we did in Arizona. We had the issue with irrigation water causing outbreaks from E. coli. And the farmers go, we can't go out there and sample for E. coli, it's just too expensive. So what we did is, and what I'm gonna suggest this to, is we did collect a lot of data on environmental factors, temperature, everything. And we found we could predict when there was a high level of E. coli based on air temperature, because it was related to water temperature, turbidity of the water, rainfall, and that. So we put that into the machine learning, which is like AI, where you take data in it and develop up the thing that says, okay, conditions are such that you should be testing for E. coli today, because the probability of you having E. coli above your level is higher. So when we developed the app, is saying, give a recommendation of when they should sample, the risks are the greater. If you get enough data using the method and get environmental data, you could probably develop an app. So what the farmers have is an app now, and they put this data in. Actually, it's automatically fed because we have weather stations in the area. So the weather, the temperature, and the other environmental factors are in there. And that's how you can use this.
Right.
Because one of the questions, what you find out is, we've investigated several outbreaks where you get repeating cases where it's a second lake. So they're always asking, what should we have done? You know, somebody put up a sign, but they feel they're ignored. So having an app with the data you develop that you can give to people that run facilities or lake or the health department, because they could look at it and say, okay, we should maybe put out signs or take some samples.
Yeah, yeah, that's the use case that we've had most in mind, is as we develop, more and more measurements, getting that other metadata around chemical, physical parameters, other microbial parameters, as a way of developing a kind of risk index. Yeah, I think would be a really useful thing to do. I agree.
And then on that note as well, was when I was over in Australia, people going to swim in the ocean, there would be trackers on sharks to determine when a shark was coming into shore. It would basically alert everyone in that particular area to say, either get out of the water or don't go swimming in the water. And maybe that's something that you can then apply, is that if the conditions are right,
Yeah.
then based on mast cells on different carriers of phones, you could effectively put out a specific alert to a concentrated amount of people and warn them, just don't go swimming.
I love that idea. That's great. Yeah, Mike.
Yeah, I think one of the other use cases we talked about is also, physician education, that if we find that the risk is highly concentrated in space and time, physicians could be more aware to, if you see something, that looks like it could be, meningitis clustered around the spatial temporal pattern, maybe suspect
Yeah, especially if we can couple this with the rapid diagnostics and the rapid treatment methods that we're all interested in. Yeah. That's great. Well, thanks very much. Appreciate it. I have learned an incredible amount today, and I hope that everybody else has too. This has been an absolute pleasure to hear from everybody around this topic, and I hope that we can carry these conversations forward and be in communication around. I've already seen several places where I know our work is related to everybody else's. So, thanks for being part of this. Thank you for coming. I don't have anything else beyond telling you that there's a really excellent lunch out there waiting for us. And I hope we can use this time just to hang out informally. And yeah, it's been a pleasure. Thank you. He's like, I've already been taking pictures of you.
Just talk about everybody that's either presented, been part of the research, like.
Also, I have gift bags for all the presenters. So you're not coming to my conference without swag. Well, thank you. Yeah. Grab one of these.
Yeah? It's good.
You read the photo, we wouldn't understand what you're saying.
Okay, what I'd like to do is state this a bit rather than making it announcement.
Anybody that's been involved.
Can I see your initiative? I'm basically in a triangle, so I'm gonna be shooting from this way. So can I have two more people here?
Like in the gym.
Yeah. Presenters, have seats here. Actually, I need you to, okay, come over here. I promise it will make sense.
Turn to the right, I'm at it.
I know this is not trying to ask you this, but can I have you stand here, and can I have you stand here as well? And pretend this flag isn't here. The camera's gonna be over there somewhere, come on. You don't need to look at it, but, yeah, like who jumps? And I have a question, is it S or a Z?
It's got a P in it, if you really.
Clemson.
Clemson.
That's like the town of, that's like the town of Wilson, North Carolina is called Wilson. It's got a T in it. Yeah.
So who am I missing? Is this everybody?
No, you're in all these people.
Okay. This is the part where we're gonna actually, if you're athletically inclined, have a seat on the floor. I'm trying to make this interesting.
All Naegleria researchers.
So these pictures generally helps us present to all of our donors. So this is like a big part, because that's the biggest thing with our foundation is people love. Oh, you should be on the.
So Jim and I should be in the picture now.
And yeah,
Craig's gonna lay across on
we'll make sure these pictures are available and Tom takes great pictures.
Yeah.
Oh, the pressure's on now. Okay.
The pressure's on Tom.
Oh, yeah.
There's a lot of fundraising.
Okay. Now, the one gentleman who's seated in the back, you could stand.
No. What do you mean no? You should do.
I know, right? Oh, sorry.
Board member. Yes. Crazy. No, what are you doing?
Oh, I was gonna get one of just the.
No.
Don't worry, we'll make sure you get the pictures, okay? It's all good.
You got elected.
Just think of that, dude, it's gonna be funny.
Now, the gentleman in the blue sweater, can I have you tighten up and I just need to. Okay.
You guys are so quiet. You can just say Naegleria.
Don't be sneaking away. Just
Make sure these,
I'm just trying to make space.
Naegleria.
Okay to tear this place apart now, isn't it? Okay. And the gentleman in the earth tone jacket,
Yeah, and the beard, yeah, you're hidden, and, no, keep the jacket on, I couldn't see you. Okay, Mike, I can't see you, so can you stand? Don't go away. I'm just. Okay. Okay.
Neither are we.
Okay.
We're still learning.
We're all trying to figure it out.
Okay, my God, it's actually gonna work. Okay. And on three, we'll take a couple in shots, just to be sure. And hold that pose, and go. Okay, and I'm almost done. This probably can't move, can it? No, it can, good. Okay. And the lady with the coral-colored sweater, can you move a foot this direction? You're hidden too much. And come forward, there's a gap there. Is a chair in the way? Okay. And Mike, if you bridge the gap. Oh, mine's a gap. Okay. I promise you it's gonna be worth it. Okay, super cool. On three. 1, 2, and 3. Hold that. 1, 2, and 3. Check that. Fantastic. Thank you so much.
Thank you.
Sorry to make it a production.
Thanks for sitting on the. Thank you so much.
I left some here for one more shot. Could that be great?
Sounds good.
Sure. Okay. It's literally upstairs from where we are right now. And I can do it in 5 minutes. Awesome. Let's do it. Run it now. We'll have lunch.
I'll come with you in just a minute.
Oh, thank you. I get, oh, thank you.
So you're doing something.